Hello, welcome to another video from the conscientious biologist, Ben Gallagher. This one's the fourth one in the cell lesson series. This is on stem cells, which are a very specific type of undifferentiated cells. Even though this video follows on from the lesson on specialist cells and differentiation, these are kind of flipping that backwards. It's part of the GCSE specification, and as always, please do subscribe to my channel if you haven't done so already. Thank you. So let's start off by asking the key question of what are stem cells? Now, I would hope you've got some kind of vague background understanding of these, or at least heard about stem cells. But to really understand them, we need to go right back to the start of the formation of a new life. Now, of course, to form a life, certainly for humans, we need a sperm and we need an egg cell. Each one of those cells contains a nucleus, and within that nucleus is DNA. Now, each one of those cells has only 50% of the DNA from the parent that created that. So there's 50% of the father held in the sperm nucleus, 50% of the mother. Of course, when those two fuse together at fertilization, they form this, a zygote. Now, a zygote is just a fertilized egg, but of course now that one's got the 50% and the 50%, and it's put them together to make that 100%. Now, the really important thing is that that cell is undifferentiated. What that means is all of the proteins can be created. If you think back to cells 2 on differentiation, we talked about DNA being wrapped up so that it can't be read, and that's how a cell differentiates. Well, in the zygote, all of the DNA is nice and open, it's all readable, so everything can be made. That cell can become everything. Of course, that means it can become literally any cell type, but of course it has to, because every single cell in your body came from that one original zygote. So that one becomes everything. So that's a necessity. Now, that zygote is going to divide by mitosis to form the early embryo. And at 1 or 2 days, you've just got a small cluster of cells that is the embryo. It's an embryo as soon as there's kind of multiple cells. Now, these cells are all undifferentiated and can also all become all cell types. So every one of those cells in that small embryo there, you could, at this stage, pull those cells apart and each one would still function as a zygote. You could put them all into different women, different uteruses, and each one should then grow into identical clones of the original person. Okay, but so they're all totally undifferentiated. Now, you let mitosis continue, and by day 5 or 6, the embryo looks something like this one here. Now, at this stage, these cells are also undifferentiated, but they've started to move down the differentiation route. So if you take cells from this slightly later embryo, we're still only talking less than a week old, those can become almost all cell types, but they're slightly more limited. Now, it's these that we call embryonic stem cells because they come from an embryo. So essentially what we're saying is that stem cells are cells that have still got the potential to become any cell. Now, not all stem cells are like this. They're not all embryonic stem cells. You can also get another type of cell because a very small number of stem cells remain in adult tissue. So all of the tissues in your body, cardiac tissue, muscle tissue, whatever, they all contain very small numbers of stem cells. But the problem with those adult stem cells is they can only become cells of the tissue type that they are within. So if they're cells within the bone marrow, they can only become blood related cells because all of your blood cells come from your bone marrow. Okay, so they're not as undifferentiated, not as useful as embryonic stem cells, and they're called adult stem cells. So you've got embryonic stem cells and you've got adult stem cells, but what we're talking about with stem cells is cells that have the potential to become lots of different cell types. So it's pointless knowing what stem cells are unless we can actually say that they're useful for some reason. And stem cells potentially could be a massive leap forward for us as humankind in the treatment of lots and lots of really debilitating diseases and illnesses and problems that people can have. So this stem cell above me is going to help me to show that. Now, they're so useful because they can become any cell type, which means they can be used to replace damaged cells in a person's body. If you look at the picture above me now, that stem cell could be induced, which means sort of forced to become, any of these cell types, or indeed almost any cell type if you're looking at embryonic stem cells. So they could become new bone cells, muscle cells, red blood cells, nerve cells. Okay, that means they could be used to replace or repair damage that's been done to a body. So let's look at a few examples of these. This one's incredible. Okay, you could use them to form new spinal neuron cells, these are the nerves in a person's spine, to repair damage after spinal injury. Now, if most of you GCSE age, you probably wouldn't recognise this guy. This is Christopher Reeve. Christopher Reeve was an actor who played Superman in the late 70s and early 80s films, the real classic Superman films. So he was a really fit, really strong, naturally muscly guy, and he was in an awful accident and was paralysed from the neck down. Now, potentially, stem cells could have been used to repair damage in his spine, to reconnect the neurons, and potentially restore ability to walk and reverse paralysis. Now, that's still in very early days. There's been lots of experiments. There's been some success in it, restoring paralysis. But it's got a long way to go yet. But how amazing could that be if you could turn back someone's life? This is a great one, replacing pancreatic cells which aren't secreting insulin. So if you've got someone who's diabetic, type A diabetic, so their beta cells in their islets of Langerhans in their pancreas don't function properly to make proper functional insulin. They still make insulin, remember what we've talked about with proteins, but they make the wrong shaped protein, so it doesn't function as insulin. But if you can put stem cells into their pancreas that will become beta cells that could make insulin, you could effectively cure diabetes. You could restore damaged or dying retinal cells in the eye. It's the retina at the back of your eyeball that picks up the light that allows you to see. Now, there's various conditions where those cells start to die and degenerate. If you could put stem cells in that could then turn into those retinal cells, you could restore someone's eyesight, or at least maintain it and stop it from getting any worse. Last one that we'll talk about, but this is one that is quite commonly used already, is bone marrow stem cell transplants. So this can be used to treat a huge range of blood diseases and immunological systems, problems with immune systems, because bone marrow is the source, contains the stem cells for every type of blood cell. That's red blood cells, all the many different types of immunological cells, and phagocytes, neutrophils, lymphocytes, all these different things, and platelets, which are involved in blood clotting. All of those cells come from bone marrow. So if you can get bone marrow from someone and you can inject it into another person, the bone marrow stem cells will just start making new functional blood for that person. It's really useful after someone's had chemotherapy that might have wiped out their own bone marrow. They could have a bone marrow transplant so that they can then start producing cells of their own. There are problems with this. Obviously, you may already be thinking, well, hang on, a bone cell transplant, isn't there a risk of rejection of that? But we'll get to that in a moment. But really, these are just 4 examples. There's many other possibilities with stem cells. Potentially any condition caused by failed or damaged cells could be treated and permanently fixed with stem cells if it can be made to work properly. And this is the problem at the moment. It's still relatively early days in this research. It's been going on for about 20 years or so, so that may seem a long time to you, but these are very complex things. Trying to work out how to get a stem cell to differentiate into the type of cell that you want it to be is a tricky thing. But we'll talk about some of the more of the practical problems with this a little bit later on. So having said how amazingly useful these stem cells can be, and we started to talk about in the first slide where they come from, let's talk about how we can obtain stem cells, because if we're going to inject them into someone to repair their eyesight or their pancreas or whatever, we need to know where we can get these stem cells from. So let's put back up the diagram that we had on the first slide, showing how embryos form, and showing next to me here the embryonic stem cells that are contained in those embryos. Those are the cells that we want. But of course, they come from an embryo. So we're on dodgy ground here, because we're talking about using an embryo as the source of stem cells. What we're not talking about though is removing an embryo that's growing inside a woman already. So she's pregnant, we're not talking about taking that embryo out. This isn't sort of linked to abortion. What we're talking about is doing this whole process next to me, but doing it in vitro. In vitro means outside of the body. So this would be done in like a Petri dish. So you can create embryos, sort of artificial embryos, in a dish like this. As long as you've got the egg and you've got a sperm, you put the sperm on the egg. Hopefully, if they fertilize and if it's under the right conditions, it's got the right growth medium, it will do mitosis and it will turn into an embryo. Now, of course, you wouldn't just do one. You might make multiple embryos in this dish. So once you've got those multiple embryos, they can then be broken apart and the stem cells can be used. Now, that's quite a scary thing in itself. There's a lot of moral and ethical issues here. We'll talk about them in a minute, but let's just answer the question of how do we obtain stem cells? Well, this generally is the method. You have to create embryos. Now, at the moment, this isn't really done specifically for creating stem cells. What does happen is when couples have IVF, if they're struggling to have a baby, what we're talking about here is basically IVF, which you've probably heard of, but it comes up in one of the phase 2 lessons anyway. IVF is the creation of an embryo outside of the body. The IV is in vitro, meaning outside of the body. The F is fertilization, IVF, in vitro fertilization. So you're creating embryos in exactly this mechanism, so you can then hopefully get several embryos and go, oh, they all look okay. Which one looks healthy? Which one looks great? Let's take that one. And you implant that one back into the mother to make her pregnant. Okay, it's a brilliant, brilliant thing that science can do to help people have babies. However, there's generally embryos left over. And at the moment, it's those leftover embryos from IVF that would just be destroyed anyway that are used as the source for stem cells. Of course, you do have an option. We talked about adult stem cells, but adult stem cells must be extracted directly from a donating patient. So if I wanted to donate some of my adult stem cells, let's say some of my bone marrow stem cells, they'd have to be removed from my bones, which means a very large needle going into my bone, sucking out some of the bone marrow to donate to someone else. But what an amazing thing to do. Bone marrow, people who give bone marrow, absolute heroes. As are people who donate any organs, of course. So you've got these main sources, though. You can either get embryos created artificially through IVF is what's used at the moment, or you could use adult stem cells. Now we're going to talk now about an alternative to those two things that I've just said about the creation of an embryo that you could get through IVF or getting adult stem cells. I'm going to talk about something now called therapeutic cloning. Now, this is something that is internationally not allowed at the moment. Officially, this has never been done for humans. Has been done for some other creatures, but not for humans. What we're talking about really with therapeutic cloning is making a clone of someone, but only letting it get to the embryo stage so that we can use the stem cells from that embryo that is a clone of the person. Now, that might be hard to get your head around, okay? It would be the equivalent of me, if I needed stem cells, trying to make a clone embryo of myself that, if allowed to grow, would possibly grow into a clone of me. But instead of letting it grow, I just take the stem cells from that embryo to then use back in myself. It's a weird concept, but let's talk through how it works. This, of course, has to be done outside of the body. So again, it's in vitro. It's done in Petri dish or something. But what we've got, instead of that sperm over there, I'm going to replace that with the patient's cell. So the patient, me, okay, you can take one of my cells, and of course, one of my cells contains one of my nuclei, and the nucleus contains 100% of my genetic information. Now, what I need to do, step one, I need to remove the nucleus from the ovum, because I don't want that 50% nucleus from the woman who donated that ovum. So we get rid of that, that goes. Step two, now that I've got that empty ovum, I program it with my own DNA. So you take the nucleus out of my cell, or the patient's cell, and you transfer it into the ovum. So now that ovum has straight away got the 100% of DNA that it would have got if it was naturally fertilized by a sperm. I've reprogrammed it. So we've eliminated that fertilization stage and we go straight down to the zygote stage, but it's an artificial zygote. It's still an egg cell, but instead of having its own nucleus and a sperm nucleus that fuse together, we've just put a full nucleus in there. But it acts like a zygote. It's just entirely mine. So mitosis will turn it into an embryo. You harvest the stem cells for use in the patient. Okay. Now, the reason why you would do this is it ensures the stem cells won't be rejected by the patient because they're genetically their own cells. If we did this using my own cell, that embryo, the cells would essentially be genetically me. So I'd just be putting them back into my body. But of course, you do have to destroy the embryo in order to do that. Now, as weird, as amazing and science fiction-esque as this all sounds, it's a massive area of controversy in science. So there's a lot of moral and ethical dilemmas here, but there's also a lot of practical issues in just trying to get this to work. So let's just think about what morals are and what ethics are. So generally speaking, morals are linked to our own personal principles. It's what you as a person or me, what I believe is right or wrong. Ethics are the widely held rules and the reasonings of our society. So if you really think about something, you decide and you sort of put some governance over it and you decide this is what it should be, that's ethics. For each person decides whether they agree with those ethics, and that forms their morality. But essentially, we can just summarize all that. Put simply, ethics and morals deal with something, whether something is right or wrong. Now with stem cells, there's a lot of arguments to be made here. And sadly, right or wrong are often a matter of opinion, which makes for a lot of arguments. And a lot of people confuse opinion and fact, which is why they get so angry, because they think, well, I think this is my opinion, and that must be correct, so you must be wrong. And it's not a case of one person being wrong, one person being right. It's just about opinions and morality and ethics. But you do need to know about some of the main points on here. So let's just quickly whiz through these. So the main points when it comes to arguing about this is that there's destruction of an embryo, which could become a human life. So this is the same argument that goes along with abortions. If you've got an embryo, that embryo could grow into a human life. Now with abortion, it's already on its way to becoming a human life. It's inside the woman's uterus and it could grow. We're talking in stem cell production, we're talking about embryos that are in a dish. So these embryos couldn't grow into a human unless something else happened, unless they were put into a woman so that they could grow. But nonetheless, you're still talking about destroying an embryo, which is the start of a life. But of course, that leads us to our second point of at what stage does life begin? Can you class that cluster of cells that we've seen in previous diagrams as a living being? It's just a bunch of cells. Yes, it will become, in the right circumstances, a living being. But do you believe it currently is an independent life on its own? So you've got this whole concept of, well, what is life? When does life start? Does it start once you've got a nervous system and it can start to think? Does it start when it has a heart that can beat or a brain that can think? It's a massive area of opinion. We just don't know, and I'm not going to get into it. The next point is about consent and violation of human rights, because if that embryo is classed as a life, then it's classed as a human and therefore it has rights. And if you're going to destroy that embryo, you haven't got consent from that embryo to donate its cells, therefore it's in violation of human rights. Now, lots of people will go, that's crazy. How can just a cluster of cells have human rights? But technically, if it's classed as a life, it does. May seem strange to you. Maybe that's not your opinion. It's the opinion of others. And that's the whole point of this. It's weighing up these different opinions. There's a massive religious objection to things like this, to stem cell technology and genetic manipulation, because it's seen as playing God. Okay, evolution, almost all religious organizations now do accept that evolution has happened, whether they believe God created at the start and God was the original programmer and created the DNA and created all the systems. That's entirely up to you to have that belief. But if you have that belief, we're talking about humans coming along and messing with what was there, with that natural process. Plus, once you throw religion into it, you've got the concept of a soul. And are you destroying a soul by destroying an embryo? So there's a whole big debate to be had there, and a very passionate and very strong debate there for some people. Moving on to more practical reasons now. So I'm putting these in a different colour so you can separate them from the moral and ethical reasons. These are more practical ones. So the one that I mentioned before is that the exact mechanism behind cell differentiation, what makes the DNA crumple up so it can't be read, in what sequence, in what bits of DNA, is not well understood. So it's really, really hard to control. So if you put these stem cells in, we don't yet have the technology to guarantee that we can make them turn into the type of cells we want. And it would be a nightmare. Imagine if you injected stem cells into someone's brain. You're trying to cure Alzheimer's, which is something that they're looking at doing with stem cells. But if you're trying to put stem cells into someone's brain to replace the damage that's been done through the onset of Alzheimer's, but instead of becoming neural cells in the brain, they turn into bone cells, and you just create a lump of bone in someone's head. That's obviously not good. So that's something that the technology really needs to work on and get right. There's also a worry that because stem cells do quite rapid mitosis as they're changing and multiplying and differentiating, that they could become cancerous, because a cancer is just a normal cell that went wrong and started dividing too fast, formed a lump, formed a tumour, and that tumour can cause a lot of problems in the body. So there is a risk that you've got to get this stuff under control properly to make sure the stem cells can't turn cancerous. Other problems with immunological rejection. If you're putting cells from another person, another embryo, whatever, that don't genetically come from that original person, then that person's immune system's likely to reject them, and it's all been a waste of time. But of course you get around that problem with therapeutic cloning. But at present, it's pretty much internationally illegal to do cloning of humans. So therapeutic cloning for humans isn't currently allowed, which means you still have a big immunological rejection. The other major problem, of course, comes with anything, is cost. This is a very expensive thing to research, to look into, and to try and do. And even when the therapies are available, they're still probably going to be very expensive, which means there's a limited number of people, only the rich, who are going to be able to benefit from this. Okay, that's it for stem cells. So the key things really you need to know. You're likely to be asked, comes up most years, about the moral and ethical arguments, and you need to say something about, list some of the pros and cons of using stem cells. So that's really the last slide. You might also be asked about the differences between embryonic and adult stem cells, so make sure you've gone through and checked that you're absolutely happy with that. And you could be asked about therapeutic cloning and how the process of that works. That comes up again when we do genetic engineering much later on in phase 2, so we will review all that at that time. From here, carry on with the cells playlist. Look at cells 5, which focuses on the process of mitosis, which is how cells multiply. Please do subscribe to my YouTube channel if you haven't done so already. I'd also ask you to like this channel, this video, if you find it interesting. I've also got a Facebook page now, so go search for The Conscientious Biologist on Facebook. I put lots of extra little snippets and infographics on there that are just more general biology that can sort of fill out your biological knowledge, add a bit more depth to your understanding. Thank you.